Hi, today we're going to talk about the first section of Chapter 1, What is Science? Our essential question is, what role does science play in the study of life? And remember that now that we're in biology, that's what we're doing, the study of life. The prefix bio means life. So think biological, biodiversity, any term that has that prefix bio, we're talking about life. So what we're going to be studying is the study of life. Our objectives today are to identify the goals of science and to list the steps that scientists take to solve problems. So we're going to be using some standards about following multi-step processes when we carry out experiments and taking measurements, performing tasks, and then analyzing those to make explanations. Here's some vocabulary that we'll be going over today: science, observation, inference, hypothesis, controlled experiment, independent variable, dependent variable, control group, and data. So what is science? Science is an organized way to use evidence to learn about the natural world. So you have to remember that science is not just a list of facts. The goals of science are to investigate and understand the natural world, to explain what happens in our natural world, and to use those explanations to make useful predictions. Now, I'm sure many of you in previous science courses have learned some of the steps of the scientific method, and we're going to review some of the steps, and then I'm going to give you an example of an experiment to kind of practice identifying the different aspects of a controlled experiment. Now, I'm not going to number the steps because it doesn't always occur in just exactly this order, but this is a basic outline of what happens in an experiment. The first thing is somebody makes observations. And then after they observe, they may infer some things based on that observation, based on those observations. We then ask a question or state the problem. And then we form a hypothesis. And a hypothesis is often stated, well, if this happens, then this should be the result. We then set up a controlled experiment. We're going to be going over the steps of the controlled experiment in just a moment. We record our results. We analyze our results, which may mean making some type of graph so that we can share our data more easily with others. And then finally drawing conclusions. And then that's the point at which we would either accept or reject the hypothesis. Accept meaning yes, our hypothesis is correct, or reject meaning our hypothesis is not correct. So what's the difference between an observation and an inference? We'll take a look at this picture. What do you see? I see red little marks from one direction and bluish little marks from another direction. I see them come together, and I see just the blue marks leave. Now you may say, well, those are footprints. Okay, well now you're making a little bit of an inference. You're using your prior knowledge. Yes, those do look like footprints. I agree. Okay, so red footprints coming in, blue footprints coming in. They all come together in the center. And then we see just the blue footprints leave. So you might think to yourself, well, what's happened here? Maybe the animal that's leaving these red footprints, okay, we could also infer that's an animal. Maybe it flew away, and that's why we don't see the red footprints leave. Maybe the animal with the red footprints got eaten. That's why we don't see the red footprints leave. Maybe the individual with the red is still there. Maybe they haven't moved. Okay, now we're making inferences. We're using our prior knowledge to try to explain what we see, to explain our observations. Now, I want to talk about a specific experiment that actually happened a long way in the past because it's a really good example of understanding scientific method. There was a very common idea for quite some time called spontaneous generation. And your textbook talks a little bit about this as well. For instance, people would see meat, and if you leave meat sitting out too long, they would notice maggots appearing on the meat. So people say, well, maggots must come from meat. So the idea that life could arise from non-life. Well, there was a scientist named Redi that set out to disprove this idea of spontaneous generation. So his question or his problem was, where do maggots come from? So he made observations. Well, he noticed that flies land on meat and then the maggots tend to appear. He inferred that maybe the flies are actually creating the maggots. So he created a hypothesis. If flies produce maggots, then if flies cannot access meat, maggots will not appear. So he sets up his experiment. He sets up a controlled experiment. Let's focus first at the top. He's got 3 sets of jars. The first set are completely open jars with no lids. The second set are tightly sealed with lids, and the third set have a netting or a cloth covering, kind of like a cheesecloth. Notice the similarities in each setup. All 3 sets of jars have the same meat in them. They're all the same size. He put them all in the same environment for the same amount of time. Now at the bottom here, the second half, we can see the results. Well, the open jars, the flies immediately were able to access the meat. Maggots appear all over the meat. Second set, flies were not able to access the meat because those were tightly sealed jars. No maggots appear. And the third set, the flies are not able to access the meat, and instead, the maggots tend to appear on top of the netting, on top of that cheesecloth. They don't access the meat at all, so that the maggots don't appear on the meat. So let's think about the parts of Redi's experiment and see if we can identify them. What was his independent variable? What did he manipulate? What was his dependent variable? What did he measure? We're going to start the first section of Chapter 1 today, and that is, what is science? It's some pretty basics of science and some things you've probably heard in some previous science courses, so it's a little bit of review, hopefully. Our essential question is, what role does science play in the study of life? And remember, study of life, that's biology. Our objectives today are to identify the goals of science and list the steps that scientists take to solve problems. Here's some vocabulary we'll be covering. Remember also in our online classroom in Moodle to check out the online flashcards that we have posted, and be sure to use those throughout Chapter 1 to review all the new vocabulary you're learning. So what is science and what is not? Well, science is not just a list of facts. Science is an organized way of using evidence to learn about the natural world. Some of the goals of science are to investigate and understand the world, explain what happens in the natural world, and to use those explanations to make useful predictions. So science is all about always trying to understand more. Here's some basics of what we would call the scientific method. When we start off, we make observations, and based on our observations, we may infer or make inferences. And I'll review that some more in just a moment. We then ask a question or state the problem. We then form a hypothesis. If this is true, then this should be our results. We set up a controlled experiment, record our results, analyze our results, which sometimes means making a graph or some type of depiction so that we can share our results with others, and then we draw conclusions. And that at that point, we then either accept our hypothesis, saying yes, our hypothesis was correct, or we reject our hypothesis and say our hypothesis was not correct. Well, what's the difference between an observation and an inference? Well, an observation is what you see, okay? Using your senses. So it may be what you see, what you smell, what you hear, what you touch. For instance, the picture in front of you, what do you see? Think for a moment. You may say, well, I see 2 sets of footprints coming together and only 1 set of footprints leaving. Well, now you're kind of making a bit of an inference. You're using your prior knowledge that those were footprints. Then you may also think to yourself, well, why did only one set of footprints leave? You might think, oh, well, the animal, again, you're making an inference, using your prior knowledge, saying that an animal might have left those footprints. Maybe the animal with the red footprints flew away. Maybe the animal with the red footprints got eaten. Now we're making even more inferences. We're using our prior knowledge to explain our observations. You're explaining what you see in front of you based on what you already know. There was an idea for quite some time that was pretty commonly accepted called spontaneous generation. And that was the idea that life could come from non-life. So for instance, if meat was left out for too long, it was noticed that maggots appear on meat. So people said, well, maggots must arise from meat. Well, some scientists thought, well, this doesn't really make sense. But we've got to figure out a way to prove this wrong scientifically. So this one scientist named Redi set out to do just that. So his question or his problem was, where do maggots come from? Well, he made observations that flies land on meat. You leave meat sitting out too long and the flies start landing on it. And later, the maggots then appear. Well, he then was inferring that maybe the maggots actually come from flies, but he had to actually prove it with a controlled experiment. So he stated a hypothesis, well, if flies produce maggots, then if flies cannot access the meat, maggots won't appear. He then set out to set up his controlled experiment. He has 3 setups. Let's look at the top first. We've got 3 sets of jars. Okay, the first set is just an open jar. The second set are tightly sealed jars with a cap. And the third set are jars covered with a cloth netting, kind of like a cheesecloth. Everything else about the 3 sets of jars is the same. The jars are the same size, they're made of the same material. We've got the same meat in each of the jars. They're put in the same environment for the same amount of time. The bottom half here, we see our results. Well, the open jars, the flies were able to access the meat, and maggots, sure enough, appear all over the meat. The second set, tightly sealed, no flies, no maggots. So you might think, he proved it. Well, the air couldn't get to the meat either, so he needed to have, that's why he had 3 setups. Third set, the flies can't access the meat again, but the air could. But sure enough, no maggots on the meat, but he did see maggots on top of the netting. Basically, now this experiment was repeated many times by many different scientists, but basically this process was able to disprove the idea of spontaneous generation. Life does not arise from non-life. So when we can't explain where something comes from, it's probably just because we just don't understand or because it's very small and difficult to see. When flies land on meat and lay eggs, the eggs are very tiny and they're difficult to see, but those eggs in fact hatch into fly larvae, which are maggots. Let's identify the different parts of Redi's experiment. What was his independent variable? That's what the scientist manipulates. What did he change? What did he deliberately manipulate in the experiment? What was his dependent variable? What was he trying to measure? What were his controlled variables? Sometimes we call these the controls. These are the things that remain constant or the same throughout the entire experiment. And lastly, what was his conclusion? Okay, well, his independent variable were the covers. What did he manipulate or change? The covers on the jars. One set had no cover. The second set were tightly sealed. The third set had the netting. The dependent variable, what did he measure? He measured the presence of maggots. The controls, well, he had the same jars, the same meat, the same environment, the same time. We could go on, the same temperature, etc. What was his conclusion? Maggots do not come from meat. They come from flies. So he was able to accept his hypothesis. Now, sometimes in an experiment, sometimes the hypothesis ends up being rejected. So sometimes hypotheses need to be revised. So if we look at the scientific method in a little bit of a flow chart, we can see that sometimes if the final hypothesis is not supported, then we create new hypotheses with new experiments and new observations. It's a constant process. It's not just a straight down line. We don't number the scientific method and say, here's step 1, step 2, step 3. It's a process that occurs, and many times it's a process that's more circular rather than linear. Now, some other terms you'll hear are, in addition to hypothesis, is what's the difference between a hypothesis, a fact, a law, a theory? I think that's important to understand. Well, a fact is something that's you can observe. A hypothesis is a proposition that may be investigated. So sometimes you've learned that a hypothesis is an educated guess. It is kind of. I think it's more of a possible explanation. Well, a law is a description of observable phenomena. For instance, the law of gravity. We can observe gravity. We can measure gravity. A theory is an explanation based on extensive evidence. So just because your hypothesis is correct doesn't mean it immediately becomes a theory. It doesn't mean it immediately becomes a law. It takes a lot of experiments and a lot of information to back that up. And there are times where there are certain things that cannot be observed. For instance, the Big Bang. We can't go back in time with our time machine and observe the formation of our universe. So the Big Bang will always be a theory. It will never become the Big Bang law. Later in the year, we will discuss cell theory. We will discuss the theory of evolution by natural selection. This is an explanation based on lots and lots of evidence that scientists have put together, but it's impossible to observe it. I want to just give you another example of a little experiment, and we're going to try to identify some of the parts just for practice. Tommy and Chucky, they think that baby food causes freckles to disappear. They have 10 test subjects with freckled faces. To test their hypothesis, Tommy puts baby food on 5 of the babies' faces. We'll call them Group A. And water on the other 5 babies, Group B. At the end of the experiment, they measure the change in number of freckles. No change is seen in either group. What was their control group? Sometimes instead of controlled variables, we identify one group as the control group. Now, what this means is this is going to be the group that does not experience the independent variable. Sometimes we call that the experimental variable. So in this case, the control group are going to be the babies that do not receive the baby food. What's the dependent variable? What are Tommy and Chucky trying to measure? Well, they're trying to measure if there's a change in the number of freckles. Because remember, their hypothesis was that they think that baby food causes freckles to disappear. What should Tommy and Chucky's conclusion be? Well, we said that no change was seen in either group, so I think we can go back and we can reject their hypothesis. Baby food doesn't cause freckles to disappear, does it? No. All right, so that's a good example. I'm going to give you guys some more examples. Please check those out on our course page. Answer the questions below for today's lesson, and we will see you in our next lesson on Section 2 of Chapter 1.